Good evening, ma'am.
Good evening, sir.
Good evening. This is the charger. In the charger. Laptop charger. Thank you. I'll leave it, brother.
Hello, ma'am.
Sir, you can start now, sir.
Yes, ma'am. Thank you. Thank you so much. Good evening, Ajay.
Good evening, sir.
Good evening, Gana. Okay. Good evening, my dear students. You are all warm welcome to the last session of the biological class in molecular basic of inheritance. In this sessions, what we are going to discuss. Already we discussed what is meant by DNA, what is meant by RNA, what are the types of DNA, what are the types of RNA, and how the transcription takes place, and also replication, and then last one is called as translation process also, how it will be occurs. All those things already we discussed. And then in this part, what we are going to discuss, you know about that DNA fingerprinting. What is the main uses of the DNA fingerprinting, my dear friends, students? Ajay, what is the main uses for DNA fingerprint?
Sir,
DNA
identification.
Identification of?
Sir, the person, sir.
Okay. So the person, whether related, relation or not related, isn't it? Okay. So here, the first thing is DNA fingerprint we are going to discuss. So before that, when you are going to analyze the DNA fingerprint, you must know some process and technique, isn't it? Then only we cannot be able to isolate the DNA molecule directly. Okay. There are some principle and procedure also followed in the laboratory, isn't it? So what are the steps involved in the DNA fingerprinting technique? So here they have given us 9 steps. So don't forget, my dear students. First of all, collection of sample. This is very, very important one. First one. Okay, they didn't give that one. Collection of samples. The collection of sample, it may be plant or animals, tissues or blood samples or whatever it may be. Okay, which contain, which one, DNA molecule. Each and every cells, each and every living organism are made up of cells, and cell within the cells there is a nucleus is there. The nucleus inside there is a DNA molecule is there. Okay, so the collection of sample is the first one. After that, DNA extraction is also you have to do. So DNA is extraction from the particular cell. There is a cell wall is there. You have to rupture the cell wall with the help of the solution only. Sometimes it may be they may use the mechanical agitation. There is a laboratory equipment. There is a process is there. After that, what happened? That a particular nucleolus is a rupture. Inside there is a DNA molecule is there. The DNA molecule is also extracted from the cell or tissue of the living organisms. After that, the DNA, okay, we have to cut into pieces because we need a specific characteristics. For example, Okay. Any one of the disease resistance characters present the 100 chromosomes, 100 nucleotide. So you have the cutted 101. How will you? We can use it some molecular scissor. That molecular scissor is called as a restriction endonucleases enzyme. Okay. So DNA first is collection of sample. Second one is DNA extraction. Third one is cut by the restriction endonucleases. After the cutting of pieces into the fragments, and you have to select desirable characteristics or desirable fragments of the sequences. Then it apply the technique is called as gel electrophoresis. Gel electrophoresis. Gel means already in our Agarose gel. Electrophoresis is nothing but the instruments that were used in where the electric field. So first one, extraction, cutting of restriction endonucleases, and gel electrophoresis. And also after that completion of gel electrophoresis, you will get the what is the band. The band also undergoes the staining process. And staining process also, it has a specific method, is called a southern plating method. And then what are the information they are having? It may be stored in the DNA banking, and then probing, and then hybridization. Last one is called as autoradiography. So these are the steps we were followed in the DNA fingerprinting. What are the things? Once again, repeat for you: DNA extraction, cut by restriction endonucleases, gel electrophoresis, staining, southern plotting technique, banking, cropping, hybridization, and autoradiography. Okay, see that one. Next slide, you can see it. What are the simple steps they followed? See that first one, there is a test tube is there. The test tube is contain the blood samples. Why they are collecting the blood sample, my dear students? Why, Pa? Ajay, why they are collecting blood samples?
So that they can analyze the DNA, sir.
How I am asking? Blood contain what are the parts is there?
So blood, so WBC, RBC,
One more
so
is that.
plasma.
A plasma is a liquid part. In one more is the solid part is that. What is that?
So RBC, WBC.
One more. How it blood clot? Blood clotting mechanism. Without this, we cannot able to.
So fibrin.
No,
No,
one
sir.
more. I'm asking about the cell aspect. Cell aspect. Okay. What about platelets?
Yes, sir. Platelets, sir.
Yeah, very, very
Sir,
important.
oh, okay, sir. Understood, sir. Okay.
Okay.
I
So-
didn't understand the question, sir.
Okay. Once again, from this, among these three cells, blood cells, which one it has a nucleus? WBC, RBC, platelets. Among these, which one it has a nucleus?
So,
Hmm.
so WBC, sir.
Very good. So that's why they are collecting the blood sample. From the blood sample, they are collecting the white blood cell, because the white blood cell containing nucleus inside, they are divided into two types: granulocyte and agranulocyte. And granulocyte means what is that? The granules present in the cytoplasm, and agranulocyte means the absence of granules. Okay, inside there is a nucleus is there, and the nucleus is containing what is a DNA molecule. So the first one is also collection of sample. The next one, DNA from white blood cell only. So then what happened now? That we are collecting the DNA molecule. The collection of DNA molecule, after that, you have to cut the DNA molecule into fragment or pieces. With the help of the endonuclease restriction, endonuclease enzyme. So as a result, what happened, there are small fragment we will attain. After the fragmentations, that desired fragments was selected and they kept under the electrophoresis. Once again, collection of sample and also DNA from the white blood cell and cut into different pieces and fragments. Okay, then it will apply for the electrophoresis technique. During the electrophoresis, what happened? The molecular weight, according to their molecular weight, they can migrate toward the charges what they are having. So anode to cathode or cathode to anode, the molecules are moving. So during the movements, what happened? That band formation takes place. Band. The band formation also, what is that? Formation takes place in the nylon membrane, which is then incubated with the radioactive probes. Nothing but the radioactive probes is nothing but the complementary DNA with radioactive nucleotide. It is also additional one. So then it can migrate. So finally, what happened? The nylon membrane, the migration of the DNA molecules adhere. Okay, adhere on the seat. Then we will collect the seat from the gel electrophoresis. Then it can be examined under the X-rays with the help of ethidium bromide as well as the ultraviolet rays. As a result, what happened? That violet color, okay, turn into cherry red. It is the final, what is that? Analysis. After that, it may be entered into the radiography or computerized one. So what are the nitrogen bases present in it? All those things we analyze, and then it will be stored in the data of in computer in the form of what is a database. So these are the technique. Okay, so this is the simple things I have given. See that one by one. First of all, DNA extractions. The DNA extraction, how it will be, very, very important for the DNA fingerprinting. See that what are the samples they were collected from the plant or animal. See here, for DNA fingerprinting, only a small amount of tissues, already told you, tissues, okay, blood, semen, skin cell, or even follicle, root of hairs is needed. So this is very, very important. So this is very, very important. In 7 cents film. So this is very, very important. First, you should collect the blood sample or semen or skin cell or even the follicle root of hair is needed. And then typically DNA content of about 1 lakh cells or about 1 microgram is sufficient. Each and every cell it contains how many? What is the nucleotide? Is there very big nucleotide? Okay, there are so many nucleotide present in it. We need 1 microgram. This is sufficient one. Okay, okay. Then after the collection of 1 microgram sample from, if the content of the DNA is limited, DNA can be amplified. This is very, very important technique. So amplified means what is that? It make it number of copies, a single copy into thousands, millions, and billions copies by making many copies of it using one of the technique is called as polymerase chain reaction. Don't forget, my dear students, the single DNA molecules, it may be multiplied into number of copies. How many numbers? A lot, 1,000, million, and billion copies we can make it in a laboratory with the help of the technique is called as polymerase chain reaction. Okay, see that after that what happened? After that, what happened? See that DNA is cut into fragment with site recognizing restriction enzyme. Which one we need? Which one is the prior quality, superior quality is the, that quality is obtained from the nucleotide fragments. Okay, with the help of the enzyme restriction, DNA restriction enzyme, endonucleases already in our laboratory. For the restriction fragment length polymorphism analysis, with the help of the analysis technique is called as which one? So this is the technique, restriction fragment length polymorphism analysis, simply called as RFLP. Okay, the cell DNA are separately cut with the restriction enzyme. As a result, mini satellite get released. Already we studied what is my mini satellite, what is my microsatellite. More than one mini satellite united together, a microsatellite. The microsatellite more than one united together as a form of macrosatellite. Macrosatellite united together form as a entire genome of a DNA. Okay. After the formation of mini satellite, what happened here? The resulting DNA fragments are separated by gel electrophoresis. Is it noted down, my dear student? This is very, very important. First, we should collect the sample. That sample DNA must contain 1000 cells or 1 microgram. After that, it undergoes the amplification process. With the help of the technique is called as polymerase chain reactions. After the polymerization reactions finished, so we have to, what is that? Cut into fragment with the help of the restriction enzyme for restriction fragment length polymorphism analysis. After that, we will obtain which one, mini satellite. After we getting the mini satellite, enter or kept under the one of the DNA fragment separation technique is called a gel electrophoresis. Okay, see that one. So this is the DNA molecule. See that one, the entire genome molecule. Are you see that, my dear students? Ajay?
Yes, sir.
Yeah. So this is the isolated DNA molecule from the sample. These molecules cut into different species, different fragment structure with the help of the enzyme is called as restriction enzyme. This is the entire molecules. Okay, it cut into what is that? Fragment or mini satellite. From this one, this round one, we, what is that? We are going to amplify, we are going to separate the molecule. This is the gene of interest, this portion only. See here, this portion only we are going to, what is that? Analyze through the gel electrophoresis. After this, what happened? What we are going to do? See the next slide, you can easily identify. See that? This is the fragments DNA molecules. This is the entire setup of electrophoresis. Okay, this is the electrophoresis. The electrophoresis, above the electrophoresis, just like a glass plate like structure, above the glass plate like structure, there is a gel coated. Okay, gel coated, it is a white colors. Okay, gel coated. But both the side connected with the electric charge. This side is anode, this side is cathode. In this part, the gel electrophoresis, there is a small well-like structure is there. Each one, it has a specific small well-like structure. The well-like structure is filled with the sample of A and B and C in different animals or different plant sources. After this completion of the loading of sample, it kept under the, which one, electric field. Okay. After the particular times or particular minutes, what happened, the gels are migrate toward the anode to cathode or cathode to anode. During this, what happened? You will obtain the band-like structure. Band. See, these are the band-like structure. Okay. then it may be separated from the gel electrophoresis with the help of the nylon. What is a nitrocellulose or nylon? What is a paper? Okay, then it can be further process. It will be done. Okay, this is the important things, my dear student. First of all, selection of sample, DNA extraction with the help of the restriction enzymes, the cut into small pieces or fragments. After that, desired gene, it will be kept under the further process. That process is called as gel electrophoresis. Okay, see that one. So this is the extraction process. DNA cut into small fragments, then it transferred to the gel electrophoresis. We will obtain the short fragment and also incubation with the label probe, and also it is exposed to the X-rays with the help of the ethidium bromide stain. After that, you will get it a band-like structure in the DNA fingerprint. Okay, this is the process. See that one. Next, the Agarose gel electrophoresis. Ajay,
Yes, sir.
don't forget, my dear, this is very, very important. Okay, so please follow it. Okay, Agarose gel electrophoresis. This is the technique used for the separation of biomolecules, particularly DNA, RNA, sometimes it will be protein molecules also. Here, the electrophoresis means with the help of electron, we can, what is that? Separate the molecules. Which molecule we are going to separate? DNA molecule. The DNA molecule into separate into fragment in the form of band through electrophoresis. So the agarose is one of the solid support used for the electrophoresis. That's why it is called as agarose gel electrophoresis. Okay, the agarose gel electrophoresis first discovered by the name of the scientist called as Arne Tiselius. Arne Tiselius is known as the father of electrophoresis. Who is the father of electrophoresis, my dear?
Sir, Arne Tiselius.
Yes, very good. Don't forget, my dear. Okay. The electrophoresis through agarose get is the standard method for the separation of identification and purification of DNA and RNA fragments ranging in size from the few hundred to 20 kb. So maximum which method they were used for the separation of the RNA molecules or DNA molecules means agarose gel electrophoresis only. If you want to separate DNA as well as RNA in accurate form means they were used which one, you know, the agarose gel electrophoresis. How? The size become a few hundred to 20 kilo base. It's a very, very minute quantity. Okay, already you know, cell is very small one. From that cell-bound organelles, nucleus is also very small one. The nucleus inside there is a DNA molecule is also very, very small. From that one, we are going to separate a small fragment. Okay, so the fragment size should be how much, you know, 100 to 20 kilo base. Okay, okay. Then electrophoresis literally means running in the electric field. Already told you. The electrophoresis technique, they were used what is that? Literally running in the electric field one. Then only the molecule, which one it has a positive or negative charge, they can migrate towards the electric field, the positive charge. Okay, cation, they move to anion. Anion, they move to cation. Then only we will obtain the band formation. Okay, see that one. See that one. See, I will show you one more is that, then only it will be. See the next, the agarose gel electrophoresis setup. Okay, this is the gel. See the next slide, it can be easily. See that this is the correct way. Okay, see, this is the gel electrophoresis. There is a glass plate is there. The above the glass plate, there is a agarose cell is coated in this part. Here there is a small well-like structure is there. The well-like structure, we will fill it with sample, and also this side the electric field. This side is also electric field connected with the buffer solution is also there. The buffer solution connected with the cathode. This one is also connected with the anode. So the both are connected with the power sources. So when you switch on it, what will happen? Automatically the power supply, what is that? Connected, isn't it? So the sample is in this portion. What is that? So negative charge. We are charging negatively. Okay. Charging side, we are loaded. So they can migrate towards the positive. They can migrate towards the positive charge. So we will obtain the band-like structures. I will show you. So then only it will be better for your understanding. See that one? So this is a simple step we are doing. So this is considered a powerhouse. Power. Okay. Power. So power. Here there is a two outlet is there. One is positive, another one is negative. Gel electrophoresis, this is the small plate is there. Just I will draw you another, so then only it will be better for your idea. Okay, colors. Okay, see that one. See that one. This is the glass plate. Okay, so the glass plate, it contains what is that? Agarose. Okay, gel is coated on it. Just imagine that I put a dot, then only it look like this is a gel. Okay, coated, fully coated. Okay, see the coated. There is a small well formation. You note it down. This is a well. So this is called a small well-like structure. Here also small well. Okay, the well is there. In this part, the connected with the lower part, there is a buffer system is there. Here there is a buffer. Okay, it is also connected with the buffer solution. Here there is a small rod is there. It's connected with the buffer solution. These are all are called as buffer. Okay, so in this part, what is that? Okay, there is a buffer solution. This is called as buffer solution. See, this is a buffer. This is buffer. Okay, so this is called as what is that? Well. A small well is a small pit-like structure. You are filling the sample. Here we are going to fill the DNA sample. DNA samples collected from the different part. What you are going? So here we are going to fill it in this both. So here also. So, okay, here also. So we are filling the samples. Okay, so we fill the DNA samples in this part. Here the what is that? This one is called as what is that? Negative charged side. This one is called as what is that? Positive side. Positive side. This is called as positive side. So the complete setup kept into the which one the negative side. So you connected with this and positive side very well. But this one is also connected with the positive side. This is the positive. So now the switch on it. What will happen? Okay, the current flow is taking place, isn't it? During this process, what happened? The molecules present here, that molecule contain DNA. DNA, it has a positive or negative charge is there. It can migrate towards the which one cathode to anode. So I will show you there. So do you know this setup, my dear Ajay? Are you following?
Yes, sir. Yes, sir.
Okay. Migration takes place which place to which place? Minus to
Plus, sir.
yes. So the molecules move towards the cathode to anode, isn't it? So that time what happened? No, automatically the DNA samples also what happened or no, they can be created. During the time, what happened? See that I will show you the. See that. So this is the plate. Plate only I draw here, so it can be easily understand. Okay. So this is the plate. Okay. The samples also loaded already in oral board in the well. So here there is a well. Okay. Here there is a well. Sample already loaded. Okay. Here the sample already oral board DNA sample loaded. Okay. The movement will be how it will be, the band-like structure. The band-like structure. See that? The band. Okay. See the structure will be like this. The band-like structure. See that one? See, the movement will be like this. Okay. Same way to here also. Same way to here also, the movement will be like this. See that? Structure will be like this. The movement will be like this. Okay. So these are the what is the DNA molecules. They can migrate toward the electric field according to their molecular weight. It is nothing but the band. This is called as band. Okay. The band, it is not equal in manner. Band, it is not equal in manner. This is very, very important. Which band is the DNA band? Okay. It's a dark and light colors according to their things. This side, what is that? Positive. This side already in oral board, negative. So this is the loaded one, isn't it? DNA molecules here loaded. They after the after the kept in the under the electric field, what happened? The migration towards the positive to negative, negative to positive. Which one Negative to positive only. Such that time what happened? You will obtain the band-like structure. This is the band. Okay. So this is very, very important, my dear students. Okay. Okay. See that one. So this is the setup. Okay. The setup. So after that, what happened? See that? The charged molecules move with a counter-charged electrode, but the electric field is removed before it reaches the electrode. Okay. See that? When it move on from this place to here, it will not touch the electrode of the opposite side. Before touching the electrode, we can remove the, what is that? Switch of the entire system. Okay. So that is the point. But the electric field is removed before it reaches the electrode of the opposite side. The movement of charged species in an electric field gives differential mobility. Different mobility. Different mobility means already told you. Okay. Each one, it has a specific band. Some of the molecules, they can move very long distance because the molecular weight is very less. So the molecular weight is very less means it can migrate towards the pole very fastly. Okay, moderate means medium. Which one it has a high molecular weight? It can move a small distance only. Two, the sample based on their charge and the consequently resolve them. This is the very, very important. The movement of the charged particle is retarded with the addition of polymeric gel so that sufficient time is available for resolving the sample. You have to kept for this setup after the loading of this one, switch on the power. As a result, what happened? The migration started. You have to wait for some times, okay, 30 minutes or 1 hour or whatever it may be according to the molecules. They can migrate from the negative to positive before reaching the electrode. That is called as resolving. Resolving means they can migrate towards the pole. See that one. Besides, occurs the polyacrylamide gel electrophoresis. Maximum that equipment name is called as polyacrylamide gel electrophoresis, simply called as page. What is the meaning of page, my dear? Ajay.
So,
What is my page? Page is nothing but polyacrylamide gel electrophoresis.
electrophoresis.
Don't forget, my dear, this is very, very important. It's also used for the same purpose, but it is prepared for a small DNA fragments and can discriminate one base differences. This is very, very polyacrylamide gel electrophoresis, mostly used for the small DNA fragments. Okay, which temperature? Here you must note it down. The electrophoresis name, page. Okay, how much it may be easily discriminated? DNA fragment in be one base pair also. Electrophoresis is normally carried out at a room temperature, 37 degrees Celsius only. Okay. When an electric field is applied across the gel, DNA which is negatively charged at neutral pH migrate towards the anode. This is very, very important, my dear student. This is very, very important. Don't forget, this is the point. So where we loaded negatively, negative side. How it will be moved towards the electrophoretic method, electrophoresis, and towards the anode. This is the question, my dear. Which is electrophoresis page. How many? The room temperature will be 37 degrees Celsius. And it can move from negative to positive. That means negatively charged cathode to anode. So that is also a very, very important, my dear. Go to the next one. So the rate of migrations, okay, how it will be moved in the electric field, this is very, very important. There are 5 steps, 6 steps have been reported here. One is the rate of migration depends on the number of parameters. What is the meaning by parameters? Loading of sample, gel and buffer, electric field. Okay. These are all are considered getting, what is that? Number of parameters. Not only that, see that molecular size of the DNA is the one of the parameter. And agarose concentration is the second parameter. Conformation of DNA is also third parameter. Composition of electrophoresis buffer is also a fourth parameter. Gel acts as a molecular size. Gel. The agarose gel, it has a molecular size also. It has a different size of the molecules. Some of them are, it has a log 10. Some of them are, it has a log 12. Like that. The size of the molecules also differ. Okay. According to the separation of the molecule. These are the parameters. What are the parameters? Molecular size of the DNA, agarose concentration, conformation of DNA, composition of electrophoresis buffer, gel act as a molecular size. These are the parameters mostly that are used. See that one? Next slide. Migration rate mobility is inversely proportional to the log 10 size of the DNA fragment. This is also very, very important, my dear. This is also a very, very important. Take a notes, my dear. Migration rate, how it will be? The mobility of the molecule on the surface of the gel electrophoresis is inversely proportional to the log 10 size of the DNA fragment. Log 10 size. Okay. See, they have given us a small equation format. Sometimes they may ask the question. Note it down. The 1 divided by mobility, that is equal to K, that is also constant, divided by log 10 size. This is the equation. Okay. So don't forget, my dears. This is very, very important. The 1 mobility, that is equal to K log 10 size. Okay. When the molecule separations you will obtain in the form of band from the gel, that process is called as, what is that? Elution process. Elution. The separated bands of the DNA are cut out from the agarose gel and extracted from the gel piece. This step is known as elution. Okay. Don't forget, my dear student, what is meant by elution? Sometimes they may ask the question. What will be the mobility rate? You have note it down, my dear student. This is also very, very important. Okay. So the elution is also very, very important. See that next one. So this is the gel electrophoresis. So here there already know very well, but there is a well is there. Okay, 1, 2, 3, 4. The different samples. The molecules migrate towards the gel plate. So which one is the largest molecule? I already told you the molecular weight. It depends upon their molecular weight. What happened? They can migrate towards the electric field. The smallest molecule, otherwise called as the low molecular weight, it can move fast on it. The lower molecular it will be in the slow only. So this one is the highest molecular weight compared to this one. Okay. According to the molecular size, they can migrate. That's why they have given us a smallest molecules band. Here the largest molecule. These are all our largest molecules. Okay. So final what happened? This condition is called as elution. From this one, this band, if you want to separate these molecules only, if you want to separate these molecules only, taken this one. If you want to separate these molecules from the agar gel to the solution, that process is called as what is that? Elution. That process is called as what is that? Elution process. This is the elution process. Then following electrophoresis, the location of a specific DNA fragment can be located by staining or by autoradiography. Suppose if you want to separate the DNA fragments, the DNA fragments responsible for the eye color or resistance of antibiotics, resistance of the tetracyclic antibodies, or whatever it may be, any particular DNA fragments. Okay, how will you identify it with a particular location with the help of staining process by autoradiography? If you want to identify, you should staining the fragments, then only it can be visible. Okay, a particular staining process also done by the one of the method autoradiography. See, so this is the important. So see that this is the gel electrophoresis. So there is a buffer already in our board. The migration takes place from here to in this spot. Okay, or here to here, whatever it may be, how it will be better. Okay, the migration towards the anode to cathode, cathode to anode. So this is the way. Okay, the migration takes place in this way only. So the large band is here, the small band is here. See the next one, already we saw it, extraction, cutting. Okay, and DNA samples also collected, transferred to the, electrophoresis we did. After that, it transferred to the electron membrane. That membrane is nylon membrane or nitrocellulose membrane. After that, what are the band is we will obtain in this portion? It may be adhere on the surface of the nylon. Then it incubated with the labeled prop. Then it will be isolated, separated from the gel. With the help of the X-rays, we can visible the DNA fragments or fingerprints. Okay, this is the methodology. See that one? So this is the remove the gel, visualize DNA fragment. We can see it. Okay, so we can see it. This is the longer fragment. This is a shorter fragment. This is the completed the gel. Completed the. This is the one side. This is the negative side or positive side. This is the negative side. This is the positive side. The molecules move toward this way only. This way. Okay, this way only. That's why you will get it the shortest molecule. Sorry, shorter molecule, longer molecule you will get. Final what happened after the separation? Don't forget, my dear. This is very, very important. Ethidium bromide. Say once again, Ajay. Ajay.
So ethidium bromide.
Yes, ethidium bromide mostly used for the staining purpose. If you want to visible the stain, you know, agarose gel electrophoresis. The agarose is also a colorless, okay, gel. Okay, and from that we are loading the DNA molecule. the DNA also migrate on the surface of the gel. It is also a invisible manner. So how will you identify it? Means with the help of a ethidium bromide staining. Don't forget, this is so many times they were asked the question, my dear. DNA molecules kept under the ethidium bromide staining only we can visible. Otherwise, it is not visible. Okay, see that one. The molecule is non-polar and readily intercalated between base of the DNA. In the non-polar environment, between the base, its fluorescence is increased about 50 times when you are using the ethidium-bromide. So what happened, the fluorescent color will be obtained. The color will be how many times? It will be more than that, 50 times. Okay. The result, it is very, very important what we are going to discuss next point, my dear. Now, the gel electrophoresis run, DNA migrate towards the gel, isn't it? After the completion of the process, we are separating the gel from the electrophoresis equipment. The band also obtained, but we cannot able to visible. So that time we have to use the staining process by the chemical is called as ethidium bromide. So that time what happened, the visible of DNA band in the form of fluorescence, how many times it will be? 50 times. Okay, see that one. Therefore, the gel can be soaked into the diluted solution of ethidium bromide and illuminations with the ultraviolet lamp reveals the location of the DNA as a band glowing cherry red or orange color band. Don't forget, my dear, this is very, very important. Ethidium bromide color will be, how it will be, look like it. Okay, the color will be changed into glowing cherry red or orange color. This is also so many times they were asked. After the application of the ethidium bromide, the DNA look like a which color band you will obtain. They were asked in the question. I think 2019 or 2018. Okay, 19, 18, 20. These 3 times. One time they were asked the question, which chemical they were used for the staining purpose. How many times it will be increases there. That one also second point. Third one, after the ethidium bromide application, the DNA look like the band look like a color, the cherry red or orange color had been given. Okay, in the previous question paper. So here you must note it down. Ethidium bromide, fluorescent color increasing 50 times. So you will get the result will be cherry red and orange color. Which color, my dear? Ajay, which color we will obtain?
So orange.
Orange or cherry red?
Cherry red, sir.
Both. Don't forget, my dear. This is very, very important.
Yes,
just
sir.
go and check it. Yeah. Don't forget. Okay. See that one. DNA fingerprint X-ray film exposure. See that one? Already we discussed. So this is the blood sample we were collected. After that, the DNA white blood cells also we separated from this one, and DNA fragmentation also takes place with the help of the restriction endonucleases. Then it's done by the electrophoresis. After that, you will obtain the band. After the band, you will obtain the, what is that? So X-ray with the help of the, which one the ethidium bromide, the structure will be look like this one. So this is the final stage. After the ethidium bromide, what happened? You will get how? The cherry red color. So this is the entire process. Okay. So there are so many methods they were used for the DNA fingerprinting. So one is called a southern method, the western method, eastern method, and also northern method. According to their country, they were used. Southern method means, what is it? Who discovered southern method? That things we are going to discuss in this part. See that one. Southern method. Don't forget, my dear, this is also very, very important. And who discovered southern method? The name of the scientist is southern. That's why they have called as a southern plating method. The first devised by, okay, the E.M. Southern in the year 1975. Southern plating result in the transfer of DNA molecule, usually restriction fragment from an electrophoresis gel to a nitrocellulose or nylon sheet. Where to where? Electrophoresis gel plate to nitrocellulose or nylon sheet. That is called as what is that? Southern plating method was discovered by E.M. Southern in the year 1975. Referred to as a membrane in such a way that the DNA banding pattern present in the gel is reproduced on the membrane, just like a fingerprint. Okay, so what the band pattern present in the gel, it can be reproduced in the nitrocellulose or nylon sheet. That process is called as what is the fingerprinting of DNA. It has a specialized method. That method is called as Southern plotting method. It was discovered by the name of the scientist E.M. Southern in the year 1975. Okay, don't forget, my dear. So this is the another one. So this is the gel plate already we obtained. See that one? Molecule from the higher molecular weight to lower molecular. These are the gel plate we'll obtain. Okay. So after that, what they are doing? See that? Effective transfer of the fragment up to 5 kp in length take approximately 18 hours, roughly equal to the overnight. This is very, very important, my dear. How many minutes will it take to obtain in 5 kp? 5 kp length, it will take 18 hours. Likewise, each and every fragments will be obtained in 18 hours only. The setup is transferred to the reservoir containing transfer buffer. This is also already told you. Buffer. What is meant by buffer, my dear? What is meant by acid? What is meant by base? Ajay?
So,
What is meant by acid? What is meant by base? What is meant by buffer?
sir,
Go.
acid is when the pH level is more, sir.
pH is more.
So sorry, pH is less than 5.
Why are you confusing?
Sorry, sir.
Okay.
pH
Okay.
is less than 5 for this thing.
Okay.
It is more than 5, sir.
Okay.
Yes,
Okay.
more
Okay.
than
Okay. Here, just you note it down. This is very, very important, my dear. So don't forget, please. Okay. See here. So clear all those things. Just a small creations. Okay. See that he taken examples. This one is 7. The pH 7. Okay. Whatever your blood pH, my dear.
So 5.6, sir. 5.3 to 5.6.
Are you sure?
Yes, sir. Somewhere around there only, sir.
Sorry. Not correct. Please. Exactly, I need a correct pH. So this is the 7 pH, isn't it? Water molecule it contain, what is that?
So-
pH 7, isn't it?
Sir,
Yeah.
so pH of blood, it will come around 7.5, sir.
5 or 4.
Yes, sir.
Okay, less or more. Sorry, listen here. Okay, so whatever may be, the pH 7 is water. Do you understand? Water. Why? The hydrogen as well as a plus hydroxyl ions are equal. Okay, so that condition is called as what is it? 7. Okay, then only the water we can drink, isn't it? Suppose less than 7, it is called as what is it? Don't forget, my dear. Less than 7 is called as what is that? Acid. less than 7. Take an examples HCl. Okay, the HCl, what is that? 2.5 only. Okay, this one, suppose if it is more, that is called as base. That is called as, what is that? This is base. See that one? This is base. Base means, take an examples, sodium hydroxide. Okay, and potassium hydroxide. These are all are has, what is that? It has more than 7 pH. Okay, so these are all are, what is the HCl and H2SO4, H1O3, and these are all are, what is that? Acid nature. Don't forget, my dear student, this is very, very important. 7.4 means, what is that? No, slightly alkaline. The blood of pH is, what is that? 7.4 in nature. That is slightly alkaline. So the neutral means water is a neutral because hydroxyl and hydrogen ions are both are equal. So we can drink water. We cannot be able to drink acid or base because the pH will be less acid. Suppose if it is high base. Okay, both are very dangerous. It will lead to some, what is a malpractice. Okay, suppose if it is enter in our body unexpectedly, what happened? You know, it will lead to completely destroy your physical nature. Okay, are you clear, my dear? Now, Ajay, are you clear now?
Yes, sir.
So what about the pH 7?
Sir, pH of water, sir.
Okay, what about the less than 7?
Sir, acid,
More than 7?
base.
So which one is HCl contain which pH?
Sir, acid, sir, 2.5.
Okay, what about the blood pH?
Sir, 7.4 to 7.5, sir.
Okay, what about the sodium hydroxide, potassium hydroxide? These are all are considered getting known as bases only. Okay, dear. Okay, thank you. Okay, please go ahead once again. So next one is, we will finish up to this level only. So because why, you know, the buffer, so I forget that one is, you know. So when you are acid or base, already you know, isn't it, my dear? Acid or base, you know, isn't it? Ajay? Hello?
Sir,
So what is my buffer, I am asking? What is my buffer?
sir, I'm not sure, sir.
Okay, okay. Anyhow, it is nothing but the solution. Nothing but the solution. When you are adding the acid or base, the pH, it will not change. Such type of solution is called as, what is that? Buffer. What is that meaning? A particular solution is that the buffer solution, when you are adding the acid or when you are adding the base, any one of the solution in it, what happen, you know, the pH, it will not change. That type of solution is called as, what is that? Buffer solution. Okay, so this is very, very important. The buffer solution is also migrate from where to where, you know, move to the plotting paper by capillary action. Just here, one thing is I will tell you, so then only you can easily understand this part. So this one we will finish it. This part, you know, so this part. So in this both the side there is a light blue color is there. This blue color is called as buffer solution. Okay, buffer solution is also covered with the a plotting paper. This plotting paper touch with the solution as well as the gel. Here also touch with the solution as well as the gel. So this has been given in the that slide. Okay, so this is the part here. Okay, see that one. So this one, the transfer buffer from the reservoir move to the plotting paper by capillary action. Which action is followed here, you know? Capillary action only. See that one, already saw it. Okay, see this one. We will finish it up to this level. See here, here the buffer is there. This is called as the small line is there, you know. The cloth wick they told here. The cloth wick is also called as, what is that, you know? Paper, plotting paper. The plotting paper touch the gel as well as the buffer solution. The buffer solution is also migrate where into a buffer into the paper into where, you know, in the agarose gel, then it will reaches the here. Likewise, here also the molecules also move. That has been given here. So don't forget, my dear students. So today what we discussed, just imagine what is meant by DNA fingerprinting. What are the steps they were used? DNA extraction, cut the restriction endonucleases, gel electrophoresis the technique, staining, southern plating, banking and propping, hybridization and auto radiography. These are the steps that were used for the DNA fingerprinting. So this is the steps. Okay. DNA first is to be extracted. How it be extracted from the any one of the plant or animal substances or samples. How much it will be? 1 lakh cells or 1 microgram is sufficient. Then it may be amplified with the help of the technique is called as polymerase chain reaction. So, and also you will obtain the mini satellite. That mini satellite, after the obtain of mini satellite, it will be entered into the gel electrophoresis. So this is the technique. Okay, they were used. After that, what happened? The electric field, what happened? They migrate towards the pole. How it will be loaded? How it will be migrate towards the pole? Already we know a little about. So as a result, we will get obtain the band-like structures, isn't it? After the band obtained, what happened now? It will be staining process also done. After the staining process, what happened? You have to place the nitrocellulose or nylon papers. So the exact copy of the DNA fragments, it will be adhere on the surface of the nylon. So as a result, you will get the exact copy of the DNA fingerprint. After that, you have to use the one of the staining process. That staining process, what is it? You know about which one? Ethidium bromide. So the ethidium bromide is also, when you spray it, what will happen? The DNA fragmentations also we can easily visible. Then it may be examined under the X-rays. What happened? The color will be cherry red or orange in colors. Yes, very, very important one, my dears. Okay. So that process, after that, we can obtain the substances from that one. That process is called as elution. So we will see the remaining thing on the next class. Southern plating method. Who discovered southern plating method? When, where, and how it will be obtained. That things also already we discussed, isn't it? So remaining things we'll see on the next classes, my dear students. Okay. Take care. Bye. Bye. Take care. Good night. Thank you, ma'am.
Thank you, sir. Thank you, sir.
Yes, ma'am. Thank you.
Thank you, sir.
Thank you, Kanna. Bye.